here to tell you about a tool that we developed to have better understanding of viral sequence diversity and evolution. Public databases are treasure troves of sequence data, and given the small genome size of viruses, they represent an entity with one of the largest number of full genome sequences. And the ongoing evolution and diversification of viruses poses challenge to vaccine and drug design and their diagnosis and surveillance. Thus, it is crucial to understand the diversity and the dynamics of viral sequence change. And we have published, I'll just zoom in, we have published a quantitative analysis of sequence changes and demonstrated this for viruses such as HIV1 clade B and influenza A H5N1 subtype. We can study a protein alignment in the context of overlapping chimeras of choice, and by classifying patterns into four motifs. This explained in figure 1 in here. I'll just zoom in more, and then we can see that index the first rank sequence or the most prevalent sequence and identified by having the highest incidence, while the remaining rank sequences with the least amino acid difference from the index are defined as variants and organized into three motifs. So the major variant, which is the most common variant sequence. The next minor variants, which are multiple different sequences, each represents in two or more of the aligned viral sequences, and each with an incidence less than major variant. And last, unique variants, uh, each observed only once in that given position. So these are only observed only once in that normal position. And in our HIV paper, we observed a unique phenomenon which we termed as index switching, illustrated in here, figure 2. And we observed that dominance of an index was challenged by its variance, where fitness change of one or more amino acids, such as through mutations, change the incidence of a given monomer sequence across its overlapping positions, resulting in a sequence rank change and thus a motif change, such as dominance between the major variant and the prevailing index. If you look closely in here, the position 313 to 321, we can see that the monomer with an E, colored red also in here, with glutamic acid, has the highest frequency with the 41% and identified as index. And the one with the D with the aspartic acid has the 35%, and this one is classified as major. And then if we move to the next number position from 314 to 322, we can see that the one with the D became an index, which was previously a major. Index in here again means with the highest frequency. So we expected this to also observe involving the other motifs, which we demonstrated in our H5N1 paper, and just and termed this broader observation as motif switching. And we illustrated this in here, figure 3, and you can see that it's much more complex and dynamic, uh, more dynamic and more complex than we had imagined. And so to provide a better understanding of motif switches in relation to viral sequence diversity, here we present MoSwA to identify all the motifs within a submitted alignment file. MoSwA not only identifies all the alignment schema position that exhibit the motif switching, but also provides a multifaceted and extensive characterization on the switches observed. So let me briefly just talk about MoSwA workflow. The input is a MSA file, and parameters are submitted by users, such as a threshold to tag low support positions and motif to analyze. And user can analyze only one motif that the person is interested in, or can work on multiple motifs like major and unique. And default parameter in here is to analyze all the motifs. Once these are submitted, MoSwA asks for a chimer size to facilitate the quantitative measurement or for the dissection of protein sequence diversity. And for this, MoSwA utilizes DEMA library. And MoSwA outputs in total of 10 files in the case of analyzing all the motif rings. And for each motif ring, there are two JSON files produced, one with raw results, including everything, no matter if there's a change or not, while the other one is filtered only with the switches. In figure 5 in here, this one is the sample outputs. Figure 5, in here we can see that one of the outputs of MoSwA, it's a cluster format, and index consensus is built by MoSwA in order to compare and view the switches. We can easily notice the switches at the specific motif and compare it with the index consensus. And MoSwA also produces a bar graph representing the clusters and hotspots of the switches. We cluster the positions that have switches based on the chimer size, like in here, figure 6. And in figure 7, sample output of the dynamic dot plot network graph to show the interaction between the motif switches. The nodes at the top bottom represents the motif switches, while the top and bottom represents the motif switches, while the nodes lying in the middle are the positions. And since index switch positions are the most interesting ones, because such positions are possibly to be avoided at vaccine targets, given the instability, we produce a similarity variability spectrum between index and the replacing variant motif based on PAM30 pairwise alignment. Thank you.